Hello everyone, welcome to my channel CAPSA. Today we will learn about Transdermal Drug Delivery System. So today's topic is Transdermal Drug Delivery System, or also known as TDDS. Okay, so what is Transdermal Drug Delivery System? It is a painless method of delivering drug systematically by applying a drug formulation into the intact skin or healthy skin. So it's a painless method for delivering drug systematically. Onto the skin. Okay. Now, how it acts? It actually penetrates when we apply the patches, transdermal patches, onto the skin. If this is the skin, this is the patches. The drugs penetrates through the stratum corneum layer of the skin and then passes through the deeper epidermis and dermis without accumulation of the dermal layer. How it passes? First, from stratum corneum, it goes to the deeper epidermis part. Then from deeper epidermis, it goes to the dermis part. Okay. Now, what are the examples? So, what are the examples? So, examples are means which kind of drugs we use in transdermal drug delivery system. That are first is nitroglycerin, fentanyl, nicotine, etc. Okay. Now, to understand the transdermal drug delivery system, we need to have a quick look on the layer of skin. So, what are the layers of skin? First layer is outermost layer. Also known as epidermis. This epidermis layer actually consists stratum corneum. Actually, stratum corneum is the most superficial layer of epidermis part. Okay. Now, the second layer is middle layer. That is known as dermis. And the third layer is innermost layer. This layer is known as hypodermis or the subcutaneous layer. Okay. Now, what are the types? What are the types? Okay. So, the types are, it is mainly three types. Okay. First one is reservoir system. Second one is matrix system. with rate controlling membrane. And the last one is matrix system without rate controlling membrane. Okay. Now, what are the polymers used for matrix system? So, what are the polymers used? Polymers mainly used are two types. That is natural polymer. One is natural and second one is synthetic. So, which type of natural polymers are used? That are cellulose, gene, gelatin and cellular. But synthetic polymers are silicon, nitrile, polyxylogen, polyxylogen. Okay, so these are the polymers used. Now we need to understand how the drugs penetrate through the skin. How drugs penetrate through the skin. Actually, there are two pathways for the penetration through the skin. So these pathways are, first one is transepidermal. And the second one is transappendigeal. Okay. Now this transepidermal layer, actually in this penetration of molecules through the stratum corneum occurs. And it is again subdivided into two categories. One is intra and the second one is inter. Okay. So in the intracellular, okay, intracellular. That is the complete of the word. So in the intracellular, what it happens? In this intracellular route, drugs actually penetrates through the corneocytes. Okay. Or transportation of hydrophilic or polar solutes occur. So, in this intracellular route, drugs penetrates through the stratum corneum cells, that are corneocytes. Okay, and in this route, hydrophilic or non-polar substances are polar substances, hydrophilic or polar substances are transported. And in the intercellular route, transportation of lipophilic or non-polar substances occur. And drugs actually penetrates through lipid matrix or here hydrophobic or polar drugs or substances are transported. In the transappendigeal route, actually, the passage of molecules through the sweat gland and across the hair follicle occurs. So, in the transappendigeal route, drugs transportation through the sweat glands or hair follicles. Now, we need to keep in mind for the developing of transdermal drug delivery system, kinetics of skin penetration is very much important. So, we need to understand the kinetics of skin penetration. So, in the kinetic of skin penetration occurs actually through the percutaneous absorption process. So, it occurs through the percutaneous absorption process. In this percutaneous absorption process, it is actually the penetration of substance into various layers of the skin and permeation across the skin into the systemic circulation. Now, how it occurs? It actually occurs through the following steps, like in the first step, penetration process occur or entry of drug. So, the entry of drug from in the stratum corneum layer or the particular layer which we are using. Okay. Now, the second step is partitioning from stratum corneum to the deeper layer. Partitioning from the stratum corneum into the deeper layer. Deeper, actually aqueous epidermis layer. Now, in the third step, what happened? Diffusion process occurred. Diffusion through the viable epidermis or the aqueous epidermis layer and to the upper epidermis layer. Okay. So, here diffusion occurred from aqueous epidermis to the outer epidermis. Okay. In the fourth point, what is happened? Penetration or permeation of the molecules from one layer into the another. Okay. So, permeation of molecules from one layer into the another. And the last step, absorption. The uptake of the drug substance into the systemic circulation. So, next is absorption. where drug substances goes into systemic circulation. Okay. Now, what are the components? Okay. So next is components. Components of TDDS. There are mainly 5 components used. First one is an impermeable backing membrane. Second one is drug reservoir. Third one is a semi-permeable membrane that actually serve as a rate limiting membrane. As a rate limiting membrane. And the fourth one is an adhesive layer. The fifth one is release liner. Release liner is a protection for the patch during storage, which we should remove before the application. Okay. Now, what are the penetration enhancer used for the penetration through the skin? Okay. Which type of penetration enhancers are used? So, penetration enhancers are of 2 types. First one is the physical penetration enhancer. What they do? They actually controls the rate. Okay. So, which kind of physical penetration use? These are actually methods like, as for example, iontophoresis, electroporation, sonophoresis, etc. Now, what are the chemical enhancers used? Chemical enhancers actually act by interacting with protein structure, or it can alter the structure of lipid, or it can improve the partition of drug. Okay. So, the compounds which alters the structure of lipid. So, first one is alteration of lipid structure. Which kind of compounds actually alter the structure of lipid? These are alcohols, sodium lauryl sulfate, or SLS, benzalkonium chloride, etc. And the substance which interact with protein. So, interact These are cyclodextran, amides, kind of pyrrolidons, etc. And the last in the penetration enhancers, which improves the partition of drug. So the last one improves the partition of these are dimethyl sulfoxide or DMSO and the propylene glycols. Okay. Now, what should be the ideal properties of penetration enhancers? So, the ideal properties of penetration enhancers. So, the first property is it should be odorless and colorless. So, the first one is odorless and colorless. And the second one is it should be specific in its mode of action. And the third one is it should be pharmacologically inert. Okay. So, pharmacologically inert. And it must be compatible with drugs and other excipients used. And it should be chemically and physically stable. So, should be chemically and physically stable. And the last one is non-allergic, should be non-allergic, non-irritant and non-toxic. Non-allergic, okay. Now, what are the formulation approach used for development of transdermal drug delivery system? So, what are the formulation approach there are mainly 4 approaches are used. So the 4 approaches are, first one is membrane permeation control system. Okay. Second one is adhesive dispersion type system. Third one is matrix diffusion control system. And the fourth one is, or the last is, micro reservoir type or micro sealed dissolution controlled system. Okay, now the last is, what are the advantages of transdermal drug delivery system? So first is, so first advantage is it provides constant plasma levels. Second one is prolonged duration of action. Third one is decreases the dosing frequency. And the fourth one is provide flexibility in the routes of drug administration and withdrawal. Or the eliminating the patches from the skin. Now, what are the disadvantages? So the next is disadvantages. That is the last part of this lecture. It shows the low permeability sometimes. So obviously it causes limitation of number of drug. So limitation number of drugs. The second one is it causes local edema, itching. Local edema, itching. Third is difficulties in large scale production. And the last is disruption of stratum corneum, which causes disintegrity of the lipids, or it can source to polymorphic transition. So disruption of lipid structure leads to polymorphic transition. So this is the end of my lecture. If you like my video, please do like, comment and subscribe and share with your friends. Thank you for your attention.